When Mendel completed his monohybrid crosses, he saw that 3/4 of the offspring in the F2 generation had the dominant phenotype, and about 1/4 had the recessive phenotype. And according to the law of segregation, and now looking at the little Punnett square that we've done, those 3 dominant phenotype individuals, if his law was correct, 2 out of those 3 would be heterozygous and 1 out of the 3 would be homozygous dominant. But by looking at them, you can't tell whether a dominant phenotype individual is homozygous dominant or heterozygous in genotype. And so Mendel set up what we call the test cross to answer that question, to verify really whether 2 of his 3 dominant phenotype individuals were actually heterozygous. And so a test cross is simply a cross of could be an unknown dominant phenotype individual, unknown genotype, crossed to a completely recessive individual. And then by looking at the offspring of that cross, you can determine what the dominant parent genotype actually is. So if the dominant parent is homozygous dominant, then when you cross that individual to a homozygous recessive individual, all the offspring will be heterozygous and they'll all look dominant in phenotype. However, if you cross a heterozygous individual with a homozygous recessive individual, some of the offspring will be heterozygous and look dominant for the phenotype, but some will be recessive and look recessive for the phenotype. Some will be homozygous recessive. If you get that result, then you know that the dominant phenotype parent was actually heterozygous. So Mendel carried this out, and he actually found that for every 2 individuals that turned out to be heterozygous, 1 would turn out to be homozygous. So he saw the correct ratio in the of the test cross results, and so that verified his idea. Incomplete dominance is a special case. Most things, most things so far that we've looked at will follow a rule of true Mendelian dominance. But incomplete dominance is a special case where you have a heterozygous individual that actually looks different from either one of the homozygous parents. Okay, so this is a special case. Typically the example that's given is found in snapdragon flowers, where the red flower is the homozygous dominant, the red is the dominant allele, and the white flowers are the recessive allele. And if you cross the red flower plant with a homozygous red flower plant with a homozygous white flower plant, you'll actually get pink flowers. So the heterozygous is pink. And we see another example here with eggplant. You have the dark purple eggplant. There's also a white allele for eggplant. If you cross a, and of course you're not just crossing the fruit, but if you cross a plant that makes the dark purple eggplant and you cross with a plant that makes the white eggplant, then you'll get actually hybrid or heterozygous offspring that are kind of a light purple. And that light purple is sort of intermediate between the dark and the white. All right, which if you think about Mendel and his kind of belief in blending initially, this appears to be blending because you have dark purple and white makes light purple. But it's not truly blending, because when you carry out the next cross, if you cross 2 plants that are both heterozygous to each other, then you get the fully homozygous dominant dark purple eggplant producing plant, and you get 2 plants that make light purple eggplant, and you get 1 plant that produces the white eggplant. So this is not blending, because even though the first part, this initial cross to this light purple offspring, seems like blending, if it really was blending, then when you cross 2 light purple plants, you should still get light purple offspring. But because this is not blending, because the alleles are separate and they segregate according to Mendels Law, you can still get back to a fully dark purple or a fully white offspring in this case. And then there's some spots, horse spotting patterns that are also incompletely dominant. So it's a limited number of examples of things that have incomplete dominance, but it is an exception to the full Mendelian dominance, the dominance that Mendel saw, where homozygotes dominance and heterozygotes looked exactly the same. I would skip this slide. I think I need to take it out. It's probably more confusing than anything. So skip that. Okay, let's look at the dihybrid cross. And we've talked about this in lab. Let's just review it pretty quickly. Mendel started with plants that were true breeding or homozygous, but now he's going to track two traits at once. There is nothing inherently different about a dihybrid cross than what happened in the monohybrid cross. It's just that now he's tracking two traits simultaneously. So if you look at, of course, the round is the dominant and yellow seed is dominant. So they're showing homozygous yellow, a plant, cross to a homozygous green, homozygous wrinkled plant. And of course the gametes that could be produced are shown here. And since there's really only one kind of gamete genotype, it's pretty simple. Fertilization will always produce a big R, little y offspring, which looks round and yellow. It looks the same as this parent, but it's not a true breeding or a homozygous anymore. It's the hybrid or heterozygous. And we can call this a dihybrid because there's two genes, seed color and seed shape, that are being considered. And this individual is heterozygous for both traits. So when this individual's eventually diploid cells make gametes, there's different allele combinations you can get in the gametes. So you have to have a representation of all the possible gamete types, big R, big Y, little y, big R, little y, little r, big Y. And so I suggest what I call a foil, maybe example of how to figure out gamete genotypes. I just take the first letter of each type, so big R and big Y, and then the outside letters, big R with little y, the inside letters, little r with big Y, and the lasts would be little r, little y. So that would get you all four gamete types. That's just one concept of how to figure out all the types. But once you figure all the gamete types, you can lay them across the top and then down the side. Since this is a self-fertilization, we'll have the same kinds of gametes for the sperm as we do for the eggs. And that's what Mendel did, so that's why we teach it this way. If you had two different parents, you would figure out the gametes separately. And then inside the boxes are the fertilizations. So of course, this is just showing a representation of what kind of pea would come from the offspring plant that was generated in each case. And of course we see the 9 to 3 to 3 to 1 ratio. 9 dominant for both traits phenotypically, 3 that are dominant for the first trait, which is shape, but recessive for the second trait, which is color, 3 that are recessive for the first trait but dominant for the second trait, and then 1 that's recessive for both. But what I want you to look at is that the law of segregation, the basic law of the monohybrid cross, has not been violated here. If we look just at color, if we look at seed color, and they've arranged these so that the seed colors, all the yellow ones are on these outside ring here. And the seed color, you have 12 yellows to 4 greens. So 12 dominance seed color to 4 recessive green seed color. So 12 to 4, if you reduce that, is a 3 to 1 ratio of yellow to green. Separately, if you just look at shape, we look at the rounds. The rounds, well, the wrinkleds are here, 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 and here. So 4 wrinkleds and then 12 rounds. So 12 round to 4 wrinkled is a 3 to 1 ratio of round to wrinkled. And so again, that's a 3 to 1 ratio. So the color is segregating in a 3 to 1 and the shape is segregating according to the 3 to 1. And so each gene, seed color and seed shape, separately are following the law of segregation. But when we combine them and look at how things are being passed simultaneously, then we see the 9 to 3 to 3 to 1 ratio. So the law of segregation is still being held up, but now we're watching the segregation of two genes at the same time. On this slide, they're showing you a little bit of the mathematical way you can look at this. So big R little r cross big R little r, that cross is happening, and big Y little y cross to big Y little y, that cross is happening, each separately. So we would expect to get 3/4 round type seeds, 1/4 wrinkled type seeds, 3/4 yellow seeds, 1/4 green seeds. So the probability of being round and yellow is just 3/4 times 3/4 is 9/16, because that's the product law. Those two things are happening simultaneously, but they're not connected, they're independent events. The probability of being round and green is 3/4 times 1/4, 3/16. Wrinkled and yellow, 1/4 times 3/4 is 3/16, and wrinkled and green, 1/4 times 1/4 is 1/16. So mathematically you can come up with the same prediction of those ratios. So Mendels Law of Independent Assortment is just saying that, and I like to rename it Mendels Law of Independent Segregation, because it is the law of segregation. It's just saying that the way that one gene segregates, like big R and little r, how those segregate into gametes, is independent of how the big Y and the little y segregate into gametes. Therefore, just because you get a big R in a gamete doesn't mean you have to get a big Y. You could also get a little y. And just because you have a little r in a gamete doesn't mean you have to have a little y. You could also have a big Y. So that's why you get 4 types of gametes, because all the different combinations of R and Y will be represented, because each gene is assorting or segregating into the gametes independent of the others. The exception to this rule is linkage, and linkage is where two genes are on the same chromosome near to each other, in which case they will not segregate independently. So I will move on to human pedigrees in the next lecture segment.